Good afternoon, everyone. Thanks for participating and attending today. So I get a few slides here to tell you about BioTime. BioTime is a company undergoing a lot of significant change. So just real briefly, I'll be making some forward-looking statements. BioTime is a leading cell therapy company. Our expertise is differentiating and expanding pluripotent cells into specific cell types, and then we use those differentiated cells as our whole cell treatments. We have 3 clinical stage programs in dry AMD, spinal cord injury, and oncology. And we have, I believe, the largest patent estate in the field with over 850 cell therapy-related patents and applications. What we do is straightforward. We take pluripotent cells and we differentiate them through various lineage protocols into specific cell types. And we have retinal pigment epithelial cells, oligodendrocytes, and dendritic cells as our 3 currently active clinical stage programs. We also do all of our own banking. We make all of our own clinical trial material. We have a GMP facility that does our scale up and moving from 2-dimensional to 3-dimensional growth of our cells. This is what our pipeline looks like. The aspect of the pipeline that I would orient you to is we have very meaningful partnerships with external funding. The Israeli Innovation Authority has supported our dry AMD program north of $12 million. CIRM has supported our spinal cord injury program north of $14 million. And Cancer Research UK supports our non-small cell lung cancer program. They actually run manufacturing and they run the clinical trial. We have an option to buy the data back for less than $2 million. Company, I said, has undergone a lot of change. There's a new CEO there, I heard. I've been on board for 6 months. We also have a new CFO from a couple of months ago. We acquired a public company called Asterius that brought us 2 of those programs that I showed you. We spun off a lot of our early stage programs and converted those into cash to develop our more advanced programs. And we also have an affiliate company which announced some very positive news recently. So this is the change that I'm talking about. Before I started, BioTime owned 35 to 40% of these 3 companies. And if you look to the right of the panel now, you can see that we own 100% of Asterius. We own just a minor amount of Ajax. We still own about 28% of Oncocyte. So the company is well capitalized. We have about $30 million of cash at our last quarter. We have yet to collect all of the payment for that early stage spin-out that we completed. And the value of our other assets is north of $60 million of equity positions in other companies. So we're about a 200 to 250 million market cap company, and a lot of milestones, which I won't go through because probably you'll ask me that question at some point during this presentation.
Well, great. Well, let's start with your RPE cells. You call it OpRegen, currently being evaluated in a phase 1/2A for dry AMD. Talk about the types of patients that you've enrolled thus far, and I know that along the way you've been presenting interim data. Talk about what you presented thus far, and you can save what you're going to present at ARVO for a couple of weeks from now.
Yeah, we were going to save it for ARVO, but why don't I just blurt it all out now? No, of course I'm teasing. So we have an active study right now. It's a 24-patient phase 1/2 clinical trial. It's intended initially to be a safety study. The first 12 of the 24 patients are legally blind. And so the curious thing, I mean, that's obviously a safety, first do no harm. But what's kind of limiting about treating patients who are legally blind is that if you're introducing new retina cells to those individuals, the only thing you can really measure are anatomical changes. So it's a safety study, but we can look for things like, are the cells present? Are they durable? Do they do retina cell things like clear drusen? But what we cannot do in those 12 patients is we can't assess any changes in their visual acuity. We can't measure things like low light reading or reading speed. So we're really excited because those 12 patients are now complete. They're on study, they continue on follow-up. But we've just recently gone into the second half of this study. These are individuals who have vision as good as 20/65. So these are people who could drive a car. These are individuals who more closely represent our intended patient population. So these are people from whom we can collect we can collect things like best corrected visual acuity, we can collect reading speed and other standard metrics of vision. And so we will have an update at ARVO, which is, I believe, May 2nd, and we'll be able to report the maturation of the data from both the first 12 patients plus those 3 better sighted patients. And that's what this story will be. We'll be watching these data as they roll out, as we go from 3 patients with 3 months data to 6 patients with 6 months data and 12 patients with 12 months data. That, we believe, will be the conviction story as that study continues.
Well, great. And I know that, again, do no harm is of key importance. And people look at your enrollment or pace of enrollment, but talk about the safety safeguards, if you will, that are part of the design that make that enrollment a little slower than maybe some people would like to see.
Yeah, so one of the safeguards, it's kind of like a belt and suspenders approach, is the patients are on immunosuppressive therapy. Even though probably most of us in this room know that the eye is a compartment that has immunoprivilege, in an abundance of caution, we maintain the patients on immunosuppressive therapy. That in and of itself wouldn't be rate limiting to enrollment, but it is illustrative of the kind of safeguards that you want to have in a phase 1 safety study. With respect to enrollment, and maybe we'll get to this, we actually are looking to introduce a new way of administering the cells to the eye, and we're hopeful that the remaining 9 patients on our study will be treated with a new way of delivering the cells that we think is going to provide some real advantages compared to the current standard of care.
Yeah, so Orbit, which you're referring to, they're here presented earlier. Talk about their device and how that approach is different than the subretinal surgery that you're currently employing.
Yeah, so what Orbit offers is a subretinal delivery system. So this is a solution for companies that are looking to deliver cells subretinally. So the challenge with the current standard of care is that the way that this procedure is conducted is that a needle is inserted into the eye, the vitreous, that gel-like substance in the eye, is removed, the needle continues through and will actually puncture the retina at the back of the eye. The cells, or whatever material you are administering at that time, this, in our case, cells, those retina cells are then injected and they form a bleb or a blister, and that is underneath the retina. And the problem is that when you remove the needle, you also can find that the cells that you just administered or just injected can efflux back out into the vitreal space. So there are 2 major problems with that. One is those cells can go float off to other places and begin to proliferate and can lead to adverse events. So that's one limitation of the current standard of care. The other problem that you can have with this is that when you pull that needle out and those cells efflux back out into the vitreal space, it means you don't know how much material you injected. So if you loaded 100,000 cells, as an example, and then you pull out the needle and you see this puff of cells come out of this blister that you just, or this bleb that you just formed, you don't know how many remained. So you have really inadequate dose control. And that has serious implications for designing larger clinical trials because that variability feeds directly into your statistical plan and the number of patients you ultimately need to enroll in order to show conviction that your treatment offers an advantage. So what Orbit has is that they have this subretinal delivery system, which is a flexible cannula. And what they do is they have, let's think of it as a needle within a sheath. The sheath goes suprachoroidally. So it does not go straight through the front of the eye. You do not remove the vitreous. There's no vitrectomy involved in this procedure. You basically come around the back of the eye and up underneath the retina. And then when you inject the material, there's no hole leading, there's no doorway leading into the vitreous. And so now when you remove the needle and you remove this cannula back out of the eye, you don't have efflux into the vitreal space. So now if you've loaded 100,000 cells, you can reasonably expect that 100,000 cells are sitting right where you injected them. So maybe the advantages now are obvious. So we entered into an agreement where we can negotiate with Orbit for exclusive rights to deliver cells in dry AMD, and we think this is going to be a game changer. We think this is the future of how you do subretinal delivery, and we're really excited to be able to work with them and have access to this device.
Well, great. We're really excited to see that data at ARVO in the next couple of weeks. Moving on to the spinal cord injury program that you acquired from Asterias, the OPC1 cells. The company reported 12-month follow-up results for all the patients in that study back in January. Can you summarize the key findings there for us?
Yeah, these are wonderful. So if in the case of dry AMD, what we're doing is we're manufacturing retina cells. In the case of our spinal cord program, we're manufacturing glial cells, specifically oligodendrocyte progenitor cells. And these are some of the cells that are lost following a spinal cord injury. And so we have had 25 treatments have been administered to individuals who suffered from a spinal cord injury, and typically these are car crashes. And what we found is that 95% of them had successful engraftment. So we can see a tissue matrix as assessed by an MRI of our material is there, filling the cavitation that's created by the absent or destroyed cells. And also 95% of those individuals had at least one improvement of their motor score on one side of their body. So with one exception among 25 patients, everybody improved. So this is not a setting, spinal cord injury is not a setting where you see there's a lot of historical data for comparison. So that's what we want to be able to do next, is take this phase 1 data now and being excited that it seems like everybody did a little bit better and everybody tolerated the treatment so well. Next we want to go into a comparative study and actually demonstrate that the difference here, that our cells are leading to greater mobility and greater sensation in a patient population which, like dry AMD, has no approved alternatives.
Right. So, prior management had talked about the next study being a randomized controlled study. And given the results that you have to date against a historical database, that level of difference, if you were to achieve it in a randomized controlled study, at least your understanding, I know you're walking into this relatively new, would that be sufficient to gain approval?
Yeah, this is one of the great questions that what is clinically meaningful, right? That's the heart of the question. How much of a benefit is enough to get an approved agent? And I think that the right way to proceed is you lead with the patient, right? So you have to ask individuals, tell me what the difference is between having mobility at your elbow and mobility at your wrist. Is that important to you? And your patients can guide you. We feel abundantly confident that the aggregate results that we've seen in that study are indicative of a clinically meaningful improvement in these patients. We ultimately are going to be asked to prove that. So that's why you do these studies, is to demonstrate with certainty that you are conferring a clinically meaningful advantage. But in a patient population that has no alternative treatment, and a patient population that can tolerate this treatment really well, my feeling is that almost any benefit is going to be acceptable. So, there's other parts of this discussion. You get into pricing power and reimbursement and so forth and such. But, to my end, I'm echoing the thoughts from this individual who was on our study who said, I can type 40 words a minute now, and I used to be unable to move my shoulders. I have everything I need in life. And so that is the kind of powerful outcome that we are hopeful we'll be able to deliver through the OPC1 program.
So I know you're going to have further discussions with the FDA on the trial protocol. Once you have that defined, and I know with you now being at the helm of the company, with other things that you're looking to bring forward, is that a study that you would look to partner, or would that still be something that you would consider doing on your own?
Yeah, partnering, on one hand, it's hard to answer because until you know exactly the size of your study, you don't know the costs and the duration. So on one hand, it's a little bit hard to say definitively, but I feel very strongly that optionality is one of the most powerful forces in our industry. So you must have your programs to be in a position to be partnerable, meaning you've got to be able to show that you've got a commercially viable manufacturing capability, compelling clinical data. You want to have the ability to partner these programs. It may be a means to an end, it may be the end unto itself, but I feel very strongly that it's incumbent upon management to always make sure that you have a program that has the option to be partnered at our election, but not be required to do so, right? Be able to reserve your own optionality to fund your own way and get to further milestones and more valuable. And if you're in the driver's seat, if you've set yourself up where you can self-fund or you can partner, now you're in control and you get better economics overall for your shareholders.
Okay, well, we're out of time, so we have to stop there, but thanks so much.
I appreciate it. Thanks so much.